also in question number 1, where it asked which one is the strongest bond, right? The strongest bond is a covalent bond, then an ionic bond, then a hydrogen bond, right? And so many of you answered ionic was strongest. I was trying to investigate why that was. I know when I typed it into Google, Google told me ionic was the strongest. But all the answers were basically based off of one web page. And that one web page I could not open because it was under a paywall. So I don't know what Google was summarizing from that web page. But in my slides and in your textbook, it does say that covalent is the strongest bond. Sometimes an ionic bond can be stronger, right? But broadly speaking, it's going to go covalent, ionic, hydrogen. Right, another common theme I saw in that reflection bit was some people were confused about monomers and polymers. Wanted to do a quick recap here. Right, monomers, they are smaller molecules, right, that can be bonded together. But when they are unbonded, these small molecules, we would call them a monomer. Right? Now when we put these monomers together, when we actually bind them together, now we get a polymer. So a polymer is a large molecule made up of repeating subunits, right? These subunits might be the same small molecule over and over and over. Like for a lot of polysaccharides, for carbohydrates, we see it's glucose, 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 glucose. All right? But they might be slightly different molecules, right? For example, in our proteins, they're all very similar molecules. They're all amino acids, but we know there's 20 different kinds of amino acids. So there's slight variations there, right? So individually, all these amino acids individually are monomers. But once we combine them together via covalent bonds, now it becomes that polymer. All right? Hopefully this clears up some confusion there. My next slide kind of elaborates on this because a lot of people are really confused about carbohydrates. And I think this might stem from that in-class activity that I had you do with the ingredients list, right? And so for that activity, I really wanted you to figure out, is an ingredient a carb or a sugar? And then look at the structure. And it's by looking at the structure that you can determine what type of carbohydrate it is. So if it's just a singular sugar, that is the monomer for carbohydrates. That is a monosaccharide. All right, that's things like glucose, fructose, and galactose. If we put two of these together, and just two sugars together, sucrose is a really common one here, where you have glucose and fructose combined together. Now it's a disaccharide. Right? So if you looked at the structure, you saw the ingredient list had sucrose, you typed in sucrose, saw the structure, saw it has two sugars in it, right, or two repeating units. Right? This would be our disaccharide. Not quite a polymer yet, because we only have two combined. But once we get bigger than a disaccharide, now we would say it's a polysaccharide. And then carbohydrates in particular, these are things like starches and fibers, and they're made of many, 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 many sugar molecules bonded together over and over and over. Any questions that I can clarify further? All right. On Wednesday, we were talking about the cell, right? And we talked about all those organelles. We got through almost all of the organelles except for one. Right? We had one remaining, which we'll talk about now. They are the vacuoles. Right? Vacuoles, they are these membrane-bound compartments, and their largest role that they play is in storage. Right? In plant cells, they provide storage, also some support. In animal cells, they provide storage. So if I were to ask you what is the role of a vacuole, you would probably say storage. But there are other organisms out there that use a vacuole in other ways. So for example, algae, right? These are aquatic plant type cells, all right? They live in the water. They use their vacuole. It's a contractile vacuole, so it'll contract in on itself to expel any excess water that might have gotten into the cell, right? So too much water can be a bad thing for that cell, so it kind of expels out any excess. And then in protists, we might see things like food vacuoles. And they play a specific role in helping to break down food particles that these single-celled organisms take in. Right? So vacuoles, storage. But as with everything in biology, there's exceptions. There are exceptions here as well. Other organisms can use them for different functions. All right, we went through a lot of organelles. So my next slide here is just a review. I want you to match this function to the organelle that does it. And I'll give you a few minutes here, look through your notes, collaborate with a neighbor. All right, let's go through this list. So, and you can hold up fingers to indicate which number here so I get more people participating. This first function, where are lipids synthesized? Threes. Threes is our endoplasmic reticulum that is smooth. 3 would be correct. All right, our second one, we're producing proteins. I see 2s. Awesome. 2 ribosomes. Yep. Oh, I went backwards. There's the 2. Should pop up. All right, third one, coordinates protein synthesis. It is 1, yes. Right, it's the nucleus, contains all of our genetic information. It decides which of that information will be decoded into proteins. All right, it hydrolyzes many biomolecules. I see 6s. 6s is correct. Our lysosomes, right? They're breaking it down via hydrolysis. What about transporting materials and sorting proteins? 5s, Golgi apparatus, also correct. Think of it kind of as like a post office of the cell. Right? It's transporting materials, it's sorting materials. We just talked about this one, acting as storage. Yes, 8 would be vacuoles. All right. These membranes are used to aid with protein synthesis. I see 4s. Yes, 4s. The rough endoplasmic reticulum. Right? They're used in conjunction with those ribosomes. And our last one, aids in detoxification. Number 7 are peroxisomes. Yes. So good review. Likely on an exam you might see some version of this type of question. Either you telling me what is the organelle, or you writing in what is the function. Maybe it might be matching. Don't know yet, but this would be a great thing for me to ask. All right, so we talked about a lot of organelles, but we are not finished with organelles. We still have 2 more. These 2 organelles are in a different category. We call them semiautonomous. Right? These would be the mitochondria and the chloroplasts. I had you watch a couple of different videos that mention mitochondria and chloroplasts here. So they're semi-autonomous because they can grow and reproduce themselves because they have their own DNA. No other organelle in the cell is going to have their own DNA besides the mitochondria and the chloroplasts. Now we call them semi-autonomous and not fully autonomous because they can't completely function on their own. They rely on the cell for synthesis of a lot of their internal components. So the cell is going to provide them the raw materials so that it can grow and divide. All right, mitochondria, we find them in animal cells. Animal cells and plant cells. Chloroplasts are only in plant cells. If we look at their functions, they function very similarly. I'm just going to briefly go over how they function because Unit 2 is dedicated entirely to how they function. But mitochondria, they're carrying out cellular respiration. Right? They're taking in food particles and converting it to ATP, which is an energy currency. Right? Photosynthesis does something similar, but instead of taking in food particles, it takes in sunlight and converts it to sugars. Sugars which then can be used as a means of energy for the cell. Right? So they're both taking something in, whether that's food, whether that's sunlight, and they're both making molecules that hold energy within them. Right? That can either be ATP or different sugars. Now keep in mind, mitochondria, again, we don't just find them in animal cells, we also find them in plant cells. Think about like roots, right? They don't photosynthesize, they're below ground, they don't get sunlight. They're going to rely on these mitochondria. All right, I had you do that quick assignment that was due today, and if you haven't, get that in. There's a little bit of buffer built in on Moodle. But it mentioned the answer to this question. So I want you to review this together. Why do mitochondria and chloroplasts have their own DNA? Yeah, so essentially if you were like study using that assignment rather than studying watching the videos. No, yeah. What's up, the same way this one? Did you see the partial lunar eclipse last night? It's kind of cool. I mean, the lunar eclipse is not as cool as someone else. What? Yeah. Yeah, it was like, it was a lunar eclipse, there was a super moon, and it was, I think, a harvest moon. It's a trifecta. All right, so why do mitochondria and chloroplasts have their own DNA? I heard it in a lot of discussions here. I know you know it. Yeah, endosymbiosis, which is my next slide here, the endosymbiotic theory, right? So endosymbiosis, right, that definition literally means a symbiotic relationship, so a relationship in which both benefit, right, in which the smaller species lives inside of a larger species. And in this case, the endosymbiotic theory is basically telling us that we are theorizing that mitochondria and chloroplasts used to be their own organism, right? They used to be bacteria cells. At one point, these bacteria cells were engulfed or taken in by these ancient eukaryotic cells, right? Once it was taken in, they both kind of realized, hey, there's some benefits to hanging around here, right? The bacteria cell gets components from the eukaryotic cell that make it better to live, right? They don't have to find all of their own raw materials. The eukaryotic cell, in return, they get these organelles that produce a lot of energy. All right, so we kind of look back here in animals and fungi and protists, we think the mitochondria evolved from a purple bacterium versus a chloroplast came from a cyanobacterium. They're kind of doing their own thing until they were engulfed by this bigger cell. Before we leave these organelles, I wanted to briefly mention structure. Right? If you look at the structure of these, it's giving us some evidence for this endosymbiotic theory. If we look at our mitochondria here, we have this outer membrane, we have a space, and then we have an inner membrane. Right? So we have two membrane layers. So likely this inner membrane, right, was the original plasma membrane on these ancient mitochondria. And when it got engulfed into the larger cell, right, it surrounded that bacterium with yet another layer of plasma membrane. So I have these two layers of plasma membrane. They also have this mitochondrial matrix. All right, inside of the matrix, right, is going to be where we are going to find the DNA. They're also going to have ribosomes, so they have their own organelles, which again is providing evidence for this endosymbiotic theory. And they have lots of enzymes used in cellular respiration. Right, two membranes and our matrix. Our matrix is kind of holding all the good stuff inside. If we look at chloroplasts, again, we see two membranes, an outer and an inner membrane. And while it's not labeled here, there is still that intermembrane space. Right? So two membranes, again, supporting our theory. Now on the inside, though, we have this system of membranes. Right? If we look and isolate just one of these membranes, right, they kind of look like these discs, maybe like pancakes. Right? These would be our thylakoids. Inside of our thylakoids, there would be a space that we would call the lumen. Right? If we stack all these thylakoids together, right, so we have now a stack of pancakes, we would call it a gram. Big picture here, lots and lots of membranes. And just like with the matrix of our mitochondria, we also have the stroma of our chloroplasts. So the stroma is the fluid outside of our membrane system, outside of our thylakoids, but inside of our double membrane layer. Right? And just like the matrix, right, it contains the DNA, it contains enzymes needed for photosynthesizing, it also contains ribosomes, so it contains that organelle. Now when we get to Unit 2, we're going to look a little bit more in depth as to why this structure actually matters and the roles that these organelles play. But for now, I'm just going to end with one more slide on these organelles, which provides yet another piece of evidence for the theory of endosymbiosis. They divide via binary fission. Right? And when we look at this, we're going to use mitochondria as an example here. If we look at our mitochondria, we have our DNA in the matrix, right? But it's not in a nucleus. So again, providing evidence that it was once a bacterium cell, once a prokaryote, right? Our DNA instead is found in the nucleoids, just the region where the DNA is found. That DNA will duplicate, so now we have two sets of DNA, and then we'll go through binary fission, which essentially, if we were to take this, pinch it with our fingers until it breaks into two, is what is happening. Right? So their ability to do this, right, also is lending to that theory that they once existed on their own. Right, chloroplasts will do the same thing. I'm just using mitochondria as my example. All right, so we looked at the structures here. I want you to work together to compare and contrast the structures of a mitochondria and a chloroplast. How are they similar? How are they dissimilar? All right, looking at our structures of our semi-autonomous organelles, how are they similar? Yeah, they both have two membranes, but they both have DNA, right? It might be slightly different DNA. Yeah. They both have DNA, they both have two different membranes. Anything else? Yeah, one has the matrix, one has the stroma, but it's this fluid inside of these organelles that have the enzymes. They have the ribosomes. They have their own DNA.
They can both do binary fission.
Yeah, they can both do binary fission. What about some of those dissimilarities? How are they dissimilar? Yeah, those internal structures, chloroplasts have a ton of membrane, this huge membrane system. Mitochondria, not so much. The inner membrane might do a few folds, right? Increasing that membrane surface area, but the thylakoids, it's this whole system of membranes. Anything else dissimilar we want to mention?
The mitochondria, like if it's the cellular respiration,
Exactly, yeah. They do different things, right? So cellular respiration versus photosynthesis. All right, so far, what we have looked at with the cell is everything inside of the cell, right? We've looked at the cytoplasm, and we know the definition of cytoplasm, right? It's everything inside. It's the cytosol, it's the cytoskeleton. We talked about those two. It's the nucleus, right? Our large organelle that contains our DNA. It's all these different organelles working together in our endomembrane system. And then finally, we have those semi-autonomous organelles, the ones that can grow and reproduce by themselves. All right, this is a great review slide. We've looked at their insides, but why can cells be so different from each other? We mentioned that, a nerve cell, what we see here, is very different from other types of cells, like a cheek cell, which we see in this other image here. Why are they different, right? What causes it to be different? We know that each cell contains DNA, and that DNA is identical, meaning every cell in your body has the instructions to produce all of the molecules that our bodies need. And in human bodies, the number of proteins we can produce is up to 400,000, right? Not every cell is going to create 400,000 different proteins. Rather, each cell will create a subset of proteins. This subset of proteins that it actually creates, even though it has the ability to create them all, this subset is what we would call the proteome, right? This is what makes cells different from one another, right? The number of proteins made, or we could say the number of proteins expressed, right? So which proteins are being expressed, right? Is it a lot of enzymes? Are they a lot of fibers? What exactly is being expressed? The level of expression can be different, right? So maybe if we're thinking about intestinal cells, right, they aid in digestion. So we might expect to find many more digestive enzymes in those cells versus muscle cells, right? So we're looking at the different types of subtypes of proteins expressed, as well as if they make the same proteins, those proteins could be modified in different ways in different cells, right? So they might have different modifications to them, such as adding in some extra carbohydrates, right? Or maybe this other one doesn't have as many carbohydrates. Right, so it's this difference in proteins is how we get all those different cells in our body. Relating to the proteomes here, right? What is being expressed, what is being made? If we were to look at different cells, we might see different numbers of organelles being made according to their function. So we're going to look at a frog, and we're going to say we harvested cells from a frog from 4 different organs, right? From the muscles, pancreas, intestine, and testes. When I look at these under a microscope, right, I'm going to notice that there's variable numbers and amounts of these organelles inside of these cells, right, depending on what their function is. So I want you to tell me, for the cell type, based on the function, what organelle do you think is going to be more prominent? And as an example, I have this first line already filled in, our intestinal cells, aids in absorption and digestion. We know that the organelle, lysosome, right, helps break down complex molecules. I might see more lysosomes in our intestinal cells than other cells in our bodies. So I want you to work through the next 3 and tell me what you can find out. Oh, we're talking about a frog.
Probably not. You would say it's carbohydrate. It's not carbohydrate. Test for
Okay, I'll just say it. What about our cells? Why do you say red? What about all right, we'll start with this first one here. So our pancreatic cells is probably the trickiest one out of the three. We are synthesizing digestive enzymes. What organelle do you think is going to be the most prominent here? Ribosomes. What else could be most prominent? Well, endoplasmic reticulum. It works with the ribosome. Right? That's once it makes those proteins, it's excreted into the rough ER. There's also one more that could be more prominent in this cell. Has to do with the word digestive. It could be peroxisomes, but I would argue likely lysosomes because the peroxisomes are detoxifying. If you were digesting, slightly different. But this would also be elevated levels. Yes. All right. What about in these testes cells? You're synthesizing hormones, specifically steroids. What organelle is going to be more prominent? Yeah, it makes lipids, and steroids are a lipid. Right? So we might expect more of the smoothie on. And our last one here, we have our muscle cells, high energy requirement. What organelle will be probably really abundant? Mitochondria, yes. Producing all of that ATP that is needed. All right, we're now going to move outside of the cell. All right. So we've talked about the cytoplasm, but what surrounds the cytoplasm? Well, every cell has a cell membrane. Right? Keep that in the back of your mind. We're going to come back to it. But on the other side of that plasma membrane, they can have other structures, depending on the cell type we're looking at, plants or animals. In plants, we're going to have cell walls, right? So outside of the plasma membrane, we're going to have this meshwork of other molecules, right? Kind of woven together to form a cell wall. In animal cells, we're likely going to find the extracellular matrix, which again is outside of the plasma membrane. It's these molecules that are woven together. See here some more woven molecules together. Find it a lot in like bone and in the cartilage. Cell wall versus extracellular matrix. Let's talk about that extracellular matrix. What does it actually do? It's this meshwork of molecules, but what does it do for the cell? Well, it can do a variety of things, right? It can provide the cell with strength. It kind of reinforces the cell. It can be support. It can help with organization. Right? So sometimes it can help move cells around within our body, but also in cell signaling, so how cells might communicate with one another. And again, if we look at this image, we see different types of molecules. We see these protein fibers. We also see these polysaccharides, and they're all woven together. So strength and support, also organization and signaling. The cell wall in plants is very similar in that it provides strength and rigidity, right? And if we look at the cell wall, again, we're outside of our plasma membrane here. Right? Plasma membrane is this purple bit here, but we see a weave of different molecules. And for plants, this is likely cellulose. Remember, cellulose is a type of carbohydrate. It's a type of polysaccharide. But in cell walls and plant cells, right, this also helps with resisting compression. Right, if we were to look at a plant cell, they're going to have a really large vacuole compared to animal cells, because this vacuole will hold a lot of water. When this vacuole is full, that's awesome, right? The plasma membrane is right up against the cell wall, right? It's a happy cell. Now say this is a plant cell in a house plant, you forgot to water it for a few days, it's starting to look a little wilty, all right? We're losing water out of the vacuole. The plasma membrane itself is going to start to shrink away from that cell wall because there's not as much water to keep it full, right? But the cell wall is going to stay the same. It's not going to move. So it's helping to provide that structure. So if our plant cells get a little dehydrated, the entire plant itself is not going to immediately just die. All right. Extracellular matrix, plant cell walls. How might you describe this to a non-science major? And I would encourage you to use Google or use AI resources to maybe come up with some analogies here. How would you come up with an analogy to describe this to a non-science oriented friend? And I'd be curious to see what you come up with. Plasma membrane being on it. We can make it work. Definitely. All right, how would we describe this to a non-science friend, someone who's maybe never heard this term again? What analogies could we use? Either cell walls or extra cellular matrix.
Cell walls
Yeah. It's something that surrounds another entity, right? Like a brick wall surrounding a house or scaffolding surrounding a building. I get the sense that that is the most common ChatGPT answer. What's another answer that maybe it gave you? Yeah, fence around a house. A water bottle? A water balloon. Okay. What's the plasma membrane in that one? What would the plasma membrane be in that example? We can make it work though. We could say it's like a water balloon. The inside is the cytoplasm. The water balloon itself might be the plasma membrane. And if we were to wrap a towel around that water balloon, that might be like the cell wall or the extracellular matrix. Okay. Yeah, oh, UPS store, we have the outside, that might be like a cell wall. The plasma membrane might be that area that the public is allowed into, right? We can make a lot of different analogies work here. I know in grad school, my professor always told me, think of it as a sweater, right? A sweater is woven with a lot of different fibers, just like the cell wall or the extracellular membrane has a lot of different woven molecules, and that kind of protects your body from the cold, right? I love using ChatGPT for this manner. I think that's one way that AI is really good with helping with is analogies to help you understand these concepts. All right. So we talked about what's inside the cell. We talked about what's outside of the cell membrane, right? But we haven't really talked about the cell membrane. That is what we're going to focus on for the rest of lecture today, and then again on Friday. So our cell membrane, right, surrounding our cytoplasm, it's made out of a phospholipid bilayer. So phospholipids, remember those are a type of fat, right? It has the glycerol attached to a phosphate group and 2 fatty acid tails, right? It's a bilayer, meaning there's 2 layers of phospholipids, and those hydrophobic tails, right, the ones that are nonpolar, the ones that don't like to dissolve in water, they're going to orient themselves inwards to create this bilayer. Right? Now this bilayer, it's semi-permeable, meaning it's going to allow some things in and some things out, right? But it can be a barrier for a lot of molecules. Now all cells have this cell membrane. Right? They're all going to have this phospholipid bilayer. They're all going to have these phospholipids, and we might have some proteins embedded within those layers as well. When we think of the phospholipid bilayer, it has those 2 layers, but those 2 layers are not identical. We would say they're asymmetrical. They look different on one side than they do on the other side. All right? So those 2 halves have different amounts of maybe phospholipids, but different amounts of proteins that might be embedded. Right? Some might have more proteins on the surface than the underside of that phospholipid bilayer. Right? They're not identical. They look a little different. You ever heard the word 2 leaflets? Leaflets are referring to the 2 halves of the bilayer. Right, there can be many different proteins in our bilayer. And we're going to go over the 3 most common. These proteins that kind of give these 2 different halves their asymmetricalness, right? Make them non-identical. The first that we'll talk about is a transmembrane protein, right? Just like the name implies, transmembrane is a membrane that is between both layers, right? It's embedded between both layers. Typically, it's going to open on both sides. A lot of times, this is how cellular transport will work. Okay, so they're embedded within that layer, both sides of them. Right? It's going to stick out on both sides of those phospholipid bilayers. We can have other proteins that are not totally embedded with both layers, but might be embedded in only one layer. Right? So these lipid-anchored proteins, what is happening here is that the protein will have an amino acid that is attached to a fatty acid, right? Fatty acid, just like we have in our phospholipids, right? They have 2 fatty acids, right? Now we're attaching an amino acid to a fatty acid, and it's going to blend in with the rest of those phospholipids. So we see our fatty acid, right, is integrated into our membrane. That is what is holding our protein in place. So that would be our lipid-anchored protein, only found on one side of our bilayer. The last type of protein we'll talk about is this peripheral membrane protein. Right? This is a protein that is not really integrated into the plasma membrane. It can be non-covalently bound to another protein. So in this example, it is non-covalently bound, so via hydrogen bonding or ionic binding, to this transmembrane protein. It can also be non-covalently bound to the polar region of a phospholipid. Right? But itself is not embedded within the phospholipid bilayer. And again, it's only going to be found on one side of our membrane system. So those transmembrane proteins found on both sides. Lipid anchored and peripheral membranes found on only one side. One is anchored in the phospholipid bilayer, the other is not. All right, so they're asymmetrical, they're not identical. In addition to being not identical, we can also say that they're fluid, right? They're not rigid. They're not going to stay that same way the entire time that it exists. We can have movement within our plasma membranes. So if we think about just the phospholipids themselves, right? The phospholipid can just rotate in place, right? So that phospholipid head can rotate. That phospholipid head can move laterally on the same side of the membrane, right? It can change positions. That phospholipid can also move from one side of our membrane to the other side of our membrane in what's called a flip-flop, right? This flip-flop actually needs energy in order to work because we have to bring the polar head of that phospholipid through a non-polar region, which is something that just doesn't mix well, right? So we have to use energy and we have to use an enzyme to actually have this happen. So it's not as common as the other types of movement, right? So it can just rotate in place. It can change places on one side of that phospholipid bilayer, or it can flip-flop and move completely to the other side. So this is why we call these membranes semi-fluid. They have movement associated with them. They are not rigid all of the time. Now, what makes a membrane more fluid or less fluid? A bunch of different factors. So if we look at just our phospholipid, right, it has these two fatty acid tails. The length of these fatty acid tails will also influence it. I just looked at my watch. Yes, we are out of class time. Briefly, before you leave though, before Friday, we're going to do a game on Friday, so I want you to watch a pre-lecture on Moodle that will be posted this afternoon. Should be a short lecture. There's this assignment that's not due until next Wednesday about scientific literacy. It's a little bit longer of an assignment. It's up on Moodle. You have a whole week to complete it.